Okay, hello everyone. It's my pleasure to welcome you all to this online symposium on gene regulatory networks, which is organized and sponsored by Immunai. So we are thinking here in this symposium on understanding the wiring diagram of a cell and how genes regulate each other to obtain cell type specific function. This is really an old and fundamental question in biology that is critical to understanding diseases as well as building new therapeutics. And we've seen a revolution on the biological and the technological side, where it is now possible to obtain high dimensional and high throughput measurements of single cells. And it's not just possible to observe cells, but through the development of these genome editing technologies, combined with barcoding technologies, you can now also perform really high throughput perturbations and observe how a cell responds to these perturbations. And of course, we haven't only had a revolution on the biological side, but also on the computational side. Here with like machine learning methods, making it possible to see through huge amounts of data and identifying important patterns in the data. So I think it's a really exciting time to bring together these revolutions. And I think this really promises to yield new insights into this old and fundamental question of inferring gene regulatory networks. So we have here a really exciting lineup of 3 speakers who will give us different perspectives on this very important problem. And so what I'll do now is I'll start by introducing our first speaker. So that's Fabian Theis. Fabian is director of the Institute of Computational Biology at Helmholtz Munich and of the AI unit across the Helmholtz Association. He's also a professor at the Technical University of Munich and associate faculty at the Wellcome Sanger Institute. Fabian has pioneered many machine learning methods for single-cell biology, in particular on modeling single-cell heterogeneity, states, and trajectories, and also more recently, more on the use of perturbations to really also get that gene regulation. So before I turn it over to Fabian, let me mention that you should all see a Q&A feature on your screen. So please feel free to submit questions throughout the talk. And I'll then pick from these questions after each talk for a 5-minute Q&A with the speaker. So with this, Fabian, the floor is all yours.
Thanks, Caro, for the kind introduction. Thanks, Immunai, for organizing this whole thing. And thanks, everyone, for joining and for the interest. I changed my story a little. I won't be actually speaking about perturbations today. Instead, I wanted to sort of put something in a context and really want to get your feedback, what you think about this one aspect. That's sort of the only new thing that I want to maybe show today, but I wonder what you think about that. So can you guys see my slide?
Yes, we can.
Awesome. Thanks so much for the feedback. So it's about this state and dynamics learning. And I want to sort of have a short introduction. I sort of wanted to put this briefly. We've been always in the lab asking about the understanding cellular underpinning of health and then also disease using machine learning methods. And I think it's still super fascinating. You have this cell, it takes some signals, and it decides whether to survive, differentiate, proliferate, or potentially die. It's like kind of crazy. It's like these little computers that know how to deal with all of these signals and then internal state in addition to then do something with it. And essentially what Caro was formulating is, I think, one of our holy grails in the field, in the sense that, we want to understand that little computer. We want to understand how these things from external and internal are being sort of wired together to do something. And I've been really trying a lot to build these networks and so on, based on bulk at the time as well. And we haven't been, I guess, overly successful, maybe even a whole field, at least in the bulk time. So I want to be a bit more radical and just say, you can't understand the system, you just know how it behaves under different states, changes dynamics and behavior. Maybe that's sufficient. And for that, we do have data. Nowadays, we have the single cell resolved data. So that's the cell times RNA data. But of course, it's much richer nowadays, right? So we have like all of these omics layers. We have now single cell multi-omics, right? Nature Method of the Year, 2008 or something. And then we have all of these different scales, now also spatial observations. You might have seen Aviv talking already about tissue biology and how we can analyze this. So I think, well, I won't go towards the ecosystem, but in a sense, our data lies somewhere on these intersections. And then, of course, and this was also what Caro mentioned, we have all these perturbations that I think nowadays that we have atlases come into play. So we can try to add these. So then our data would be somehow in this cube, and I just call it now X, S, so omics, scale. And I guess sort of the super abstract version, what we try to do in the lab is then just try to function how a new state is sort of being made from an old one, potentially as a function also of a perturbation. And in a sense, if you have this S, this kind of needs to contain the wiring diagram of whatever type of object you now throw at this. So some type of abstraction of this network. Of course, it might not yet be a network. So in order to do so, there's all kinds of steps needed, sparsity and whatever. And there's like, I think, very interesting modeling questions there. For now, I just want to speak a little bit about that function. And here, just like this one sketch, which I really like so much in our field, now that we have this thing as a resolution, maybe we can really understand how cells go through healthy aging, but maybe then also this turning off into diseases. And that's sort of my general, I think, motivation to understand then this state change formula, let's say across time. Maybe you have this health angle, and then you just like understand how a state change, sort of a function of the old one, and that could maybe be answered with pseudo-temporal ordering, things like that. And Sam, I'm sure, will show some additional cool ideas for that. And then I think an additional angle, of course, is then to sort of make these changes between trajectories. That's where you sort of add these perturbations. I think those two things are quite interestingly connected. Of course, the tool to analyze is going to be machine learning. And before I talk about that, I just wanted to highlight that we've been sort of investing quite a bit into infrastructure, such as now this sort of analysis framework, ScanPy, and many things downstream. So 2 points about the dynamics aspect before I speak about the general functionality. I guess in a sense, gene regulation can be seen as understanding how gene state changes under across time, maybe also across perturbation. So if you want, this F could be an approximation to a GRN. So it could be really based on a regulatory network, could be like have sparse entries and then tell you just like take only transcription factors there and then tell you about the next state. It could be maybe also using open chromatin depending on what X is. We have all these different S's there with different omics levels. But in any case, if you have that, you can do something with that. And in most cases, we've been approximating this and we didn't have the derivative and we've been sort of just working on X. But I think with ideas such as this, I would say seminal idea from RNA velocity from Linus and Kachenko labs, who essentially sort of based on ratios of unspliced and spliced reads came up with just simple RNA metabolism and were able to then estimate something about this dynamic and approximate this, you can maybe go in this direction. And there are some early ideas in that paper and some follow-ups where you essentially estimate this, for example, with linear models. We've been following this up with a fully dynamic modeling aspect, again, also though per gene only, so nothing gets connected. And I guess using this or related ideas to then build these networks, I think could be one quite exciting idea. There's some features in this SVL approach that we did at the time, and we've been then following this up. There's a bunch of new developments we're doing, which hopefully we can talk about sort of more robustly next year. But one thing I just wanted to mention is, this is sort of just the underlying kinetics picture again. So, this is RNA metabolism. Essentially, you have this modeled separately for each gene. And ideally, you want to then combine it. And the simplest way to combine is just like stack those vectors where it would go next. So you just look where they live in this spliced times unspliced space. This would give you sort of a proxy for velocity. If you stack this across all genes, you get one vector and you project it again back down to your manifold. You sort of see where one cell would be heading. So that's this idea. I think there's interesting questions, of course, going beyond global regulation rates and then adding different models and so on. So there's all kinds of ideas. We discussed this in this perspective. But the one thing that we've been doing once we have those kinetics, and this could be from RNA velocity, but could be also estimated from all kinds of other things, such as metabolic labeling, we can then actually do something with it. Once we have this net, we can do predictions. One prediction we've been doing here, this is something we call cell rank, because it sort of mimics Google's PageRank algorithm. And in a collaboration with Dana Pear's lab, Marius, the PhD student who did this, was then actually trying to estimate, to essentially integrate all of those arrows. So you have this vector field and its high dimensions, and you just like many random walks run along these arrows, and you see where they come from and where they go, and then you can sort of get terminal as well as initial states and also intermediate meta-stable states. And in particular, you can now count how many random walks go through this thing, and you can give probabilities whether they go left and right. And you can do this without actually running random walks because you know how to deal with Markov chains. So that's, I think, a somewhat useful tool, and we've been sort of building upon this quite a bit. And I think somehow in there, because essentially, again, connect directions to the state, there has to be a better encoding of the regulatory network, and potentially can access that. So that's the part about the dynamics. Now we sort of go a bit more into steady state, but a large scale level. And this is essentially about atlases now, right? And how we measure now state. So we have this manifold. Where's this manifold coming from? Well, this is, I guess, something we can get in the future from atlases. I think Human Cell Atlas is one of these big examples. There's more of these ideas around, where we essentially try to categorize different cell types into a periodic system of cells instead of elements, right? We've been doing this for long. We've been contributing essentially to both mouse and human lung cell atlas, and we've been actually now nearly wrapping up a big integrated atlas across 15 different sites to really have sort of a model for variation across labs, but then also humans. We've been using some of these data sets already, for example, here for a study where SARS-CoV-2 entry genes are being distributed across each of these lines, essentially being a humans or across clinical covariates such as age, gender, and smoking status, and indeed found some associations there. I though want to, we want to talk about GRNs and understand how cellular state is encoded in these things. If you build atlases, you somehow need to understand, at least implicitly, what the important directions are. And if you check what we wrote, the definition of Human Cell Atlas at the time, it was essentially we want to build a collection of cell reference maps. And those reference maps have to be consistent and be able to map things. And if you just do this for one data set, that's what we usually find. But if you now add many ones, then we don't actually see an ordering by cell type, but we very often see like one covariate strongly being, one axis of variation strongly being driven by study. So essentially where different places come from, let's say by patient or something like that. And if we were to find any type of causal model that underlies systematic things across human variation, it better does not be impacted by that. So one important early step is to remove this variation by, for example, data integration. And this process should then learn what are the interesting axes, the biology relevant ones versus the unrelevant ones, right? So this again contains already something about this state change. So this needs data integration, and in machine learning terms, it's just essentially latent space learning. So we've been playing around with these. Many have actually, I think, first methods particularly proposed by John Marioni's and Raul Satija's labs. But now that you have these, you can ask queries on top of that. The question is now, what sort of popular, how do these things work? So we've been setting up a challenge for that. Actually, not only for single cell data integration, for like just one modality, but across modalities. We built one of the largest data sets. This is collaboration with Smita Krishnaswamy's lab, as well as many others, to build this sort of a big data set challenge with more than 160K cells across multiple sites, and actually asking challenges of cross-modality prediction, matching cells, or learning latent spaces. And I don't want to go into this challenge, really encourage you to look at it, but the interesting point was that pretty much all of the winning models in some sense or another used neural networks. So just showing you 4 of the different models that have been proposed there. They're all based in one way or another on this concept of learning joint latent spaces. So this is what I want to now introduce in the following, and this is, I think, sort of related to what many of us nowadays use. And if you're interested in this general thing, there's sort of more challenges in this openproblems.by website. So deep learning for latent spaces. Deep learning is now obviously revolutionizing computer vision in many areas of computer science and applications as well. And in our field, it's a bit different since we don't have individual cells labels, so we often use unsupervised learning techniques. These things where we sort of squeeze down data, blow them up again. And for example, throw in sort of sparse data here, and can you, for example, use it for imputation. And in a sense, this bottleneck learns a meaningful representation of, for example, a differentiation process that was just indicated here. So in a sense, this architecture here, we're speaking about networks. It's a network that eats genes in a sense, right? So I think here, if we interpret these, there's actually an encoding of this regulatory network structure. It necessarily has to be, because it has to understand what the important directions is. Whether it directly includes that, or sort of a bit more sparsely, whether we add priors to make it include that, that's sort of ongoing discussion. Quite a few papers have been coming up recently that add one or another way of a prior, and I'll be discussing this in a minute, maybe a bit more. Let me first show you what early examples for this were. I think with the first 2 papers were SCVI from Nia's lab that had one of these autoencoder structures, actually variation one. We had at the time a normal autoencoder, but in this case, not an MSE cost function, but an adapted negative binomial cost function that we used to then just denoise. The main finding that I really like about this paper was that we actually saw that if we really go with the latent space to very low dimensions, in this case, just look at PBMC's colors indicating different cell types, but this looks pretty much like the UMAPs or TSNE we often draw. So this latent bottleneck layer, he really learns this representation I was indicating before. We've been using this then for a bunch of things. So Carl was introducing perturbations already. So this comma P and the function that we try to learn, we can actually learn with this representation. So in this case, for example, we have PBMCs and control and stimulation. Then we left out some cell type and we're actually able to predict it. In this case, by looking into latent space and learning sort of the shifts of all the other cell types and then using this one to generate this out of distribution problem. And I think in this way, we could have a potentially useful model to, in the longer run, also model perturbations robustly. What I want to now sort of put forward is that we were somewhat naive at using currently our models. And there's all kinds of constraints and invariances that we can ask on top of these networks. And here I cite from a rather nice perspective from Al-Qarashi and Sorga, who've been thinking about what they call differential biology, so essentially how to use deep learning for then modeling really mechanisms, which is a bit of an oxymoron, right? So the mechanistic model is really different from these sort of more black box neural network models. I think a lot of us have been trying to sort of go at this intersection. And one early point they made in the paper, there's all kinds of additional constraints that are naturally being put into the models nowadays, also in computer vision, many other fields, such as convolutions, right? Things this should be like a shift invariant, group equivalence, 3D molecule structures that should not be depending on space, and many others. Well, we don't usually have some of these in our models yet. Essentially, we just deal with tabular data, right? So I think adding some more of these type of constraints could actually help quite a bit. For example, we've been playing quite a lot with graph neural networks now to at least model spatial positions of cells in spatial omics, but I think there should be more to be done, particularly with regulatory structure, where there's some results have been done in bulk, but I don't think in single cells. The other point that I really liked about this paper, and I definitely encourage you to check that out, is that they've been sort of saying, traditionally, we have these principle-based type of models. In physics, when you throw a ball, where it falls, so you can write up equations. Then in conventional model, we just have these black box data-driven models. That's a little bit what I showed you before. What they propose is maybe that you can have something in between there. And they call it differential programming because it combines some principle-based small modules in a sort of more consistent fashion with intermediate statistical steps. And that's a key point, keeping everything differentiable to really build up end-to-end models. And I think that's an interesting point that we might not yet appreciate so much because we don't yet have sort of things too much integrated across layers, but I think at some point we would be hopefully also going in this direction. And now questions that directly pop up from this would be, of course, can we somehow reuse those primitives? How do we do this? Like, let's say if we have an atlas, how can we reuse and how can we add constraints? And I want to show you two vignettes for this before I wrap. So the first vignette is to reuse information. And for this, the simple idea is that, traditionally we just like have separate tasks. But if you want to now query a data, reuse sort of the reference, we need to then somehow transfer knowledge. That's essentially this reusing idea, right? And that's what we did with this SCArtist tool that came out earlier this year, proposed and led by Mohammed in the lab, where we essentially have a bunch of public reference data sets and sort of put this into this neural network architecture, but add now reference labels. We have a conditional relational autoencoder to keep track where a study is. In this case, we can clean up this latent space here. And the key trick is now, and this is sort of this reuse primitive, store this encoder here in a database and reuse this one, for example, to transfer between healthy and disease situation. So in this case now, if you have new query data, you'd be doing the same as before. You just reuse this thing from above, but now you do what we call architectural surgery. You add now additional nodes here for the queries because you want to now also add those into the rest of the structure. And then you just need to adapt these and not the whole thing. And that's why you can then map your own new data on top of the existing one without having to have access to original samples. You just need to have this primitive. And once we have this, we could potentially concatenate it with additional things that to people that don't even want to understand the whole variation and things, they just like use this thing, and then they can embed and learn the atlas. So this is something that actually works. And I do have an example here, but I think I'm going to skip this if I really just want to stick to my 20-minute point here and just show you a little bit about Outlook, what we do with these things. So this other one is in the paper, and I encourage you to check this out. This is actually a collaboration with Adam from Yosef's lab, where we've been using this things architecture surgery to actually build multimodal reference atlas. They had this nice idea of having a multimodal learn not only of RNA, but also of targeted protein on site-seq, so that they call this total VI. And they could, for example, integrate two dataset PBMCs with total VI. What we now do is wrap this total VI within this SCArtist framework, and then add a query that's only RNA, map this then on top of it by just reusing the encoder architecture from the RNA, and then actually be able, then we're able to integrate this RNA-only data and this combined data. So you're using the reference like in genome sequencing, we also just map onto existing genome and then can read off all these additional drags in the genome browser. In this case, we just read off the protein, and actually we can see that the predicted proteins match up very nicely. So this is working out. What we're ongoing doing is now starting to interpret these mappings, and I'll say a little bit towards that in a minute, but then also do classification based on these latent space features and have multi-omics extensions. So that's the first part. The second part that I wanted to have, the second vignette, is that we now add these constraints. Remember in the Karashi-Sorga paper, they proposed in differential programming that we could potentially sort of make some things a bit more principled. What's principled in our case? Well, there's a lot of sort of even basic gene regulation models. I was hoping that this SCVelo sort of basic model could be a more principled model to add. We're playing around with this. But simpler one is that we know what gene programs are, right? So we can encode prior knowledge of genes that stick together into programs. So we have a matrix here of saying essentially gene is contained in program 1, 2, 3. And it could be, for example, gene ontology, it could be reactome, things like that. Or could be also something in a purely data-driven fashion. But in any case, what we now do is we now look into our encoder, have the normal VIE as before, but now try to interpret these variables. We've been trying all kinds of things such as disentanglement and so on to make this easier to understand. But the easiest thing we came up with at the moment is now to only have a linear decoder. This way we can constrain this one directly to the output X, and this linear constraint decoder we can now add then this thing here as a constraint. So essentially just allow to train weights that are contained within one of these gene processes. And the additional trick, we have something about the masking that I don't go into detail now, but the trick that we need to do in order to now potentially have much too large bases here, we add now a group lasso layer to really just select those gene programs that you need for your particular data set. And in this way, we can then essentially really interpret, like in scatterplots, how you want each of those different gene programs that you have in this low-dimensional latent space. So in contrast to before, where this was just like a U, like in a UMAP, now this one direction really indicates activation of a gene program. And that's, I think, quite interesting to analyze data. Of course, with interpretability, big thing in explainable AI, right, comes usually a loss of prediction power. And quite interesting in our case, because maybe samples are not yet too large, a variation is maybe too subtle, we actually saw that we can use this and we don't lose that much power. Unfortunately, I'm so sorry, I forgot to, once we have this set up, of course, what we want to do now is we want to now transfer this just as we did with SC arches and now have a new data mapped on top of that now has the interpretable axis. Once we map on top of that, we can now just use our pre-trained reference and do surgery in the reconstruction, thereby allowing us to add additional new pathways here that won't contain this one. So we can also use this for gene program learning. All right. So the first interesting result that we find is that this explainable programmable map, or this XP map, does not lose expressiveness versus nonlinear models, even though we have this linear model as constraint. So for example, if we go in blood here across 8 different batches, we actually see that overall SCVI unconstrained behaves pretty much like the XP map. And if we do this more systematically across many different batches here, this is taken from one of these sort of quick to load databases from our Spira tool. I encourage you to check that out. Essentially what we find is that XP map was in parts even outperforming SCR, or at least sort of performing on par. So we don't lose expressiveness, even though we have this linear decoder, which I think is already quite fascinating. And now that we have this, we can now see what it does. And this is, I think, my second last slide before I stop, Carl. So for example, here we have now a PBMCs, a bunch of control ones, and one where we have interferon stimulation. And you see, if you have the full PBMCs, we actually see then some sort of thing here pointing out that's really specific only in the stimulation. And then we can calculate base factors, what's different there, because we have our interpretation of each of the dimensions, and we see that interferon signaling is already popping up. Then we can actually look then in the embeddings, and we see that this is consistently true. And interestingly, if we now remove our top interferon beta associated pathways, then this new embedded class is actually being removed. So it really needs this direction, those directions, and explain the perturbation. So we can use this to explain directionality and perturbation. And then I can also show that this helps us to add pathways, but I think I skipped this and I come to my conclusion. With the cell rank, we can learn about dynamics and we get some interpretation of regulatory networks. We can then sort of use these vignettes of differential biology and try to use them either transferring subparts of the models, or also using explainable matter. And I think as extension, we can of course also add perturbations in similar fashion and spatial data as well. This is the lab. Thank you very much for attention.
Thank you so much, Fabian, for a really great talk. So we already have some questions. So let me just start with one, which I think comes often up, namely the question of, how good is your performance in the latent space, right? When you are training on some data set, and you have, of course, introduced transfer learning, but like if you don't even do transfer learning, how well does actually this latent space work afterwards if you're using it on, cell types or cell states that you have not seen before in the original data set, which you use to generate the model?
It's an interesting question. Like there's so many metrics that you can actually ask, how well do you do? And I think we need to be sort of a bit aware what those things could be. And indeed, for example, for classification, it doesn't really matter all that much. So I think cell type prediction, it either works okay, and then it, even a linear method does a good job, or you have some other potential issues. I think for integration in particular, that's sort of a nice metric. And in this NeurIPS competition, we also have this multimode. That's also, I think, a nice problem, which is sort of not as straightforward. And for that, actually, just completely unconstrained latent spaces do okay. But if you add more, usually it sort of starts expanding on this. So I think we would also be sort of going through maybe this, what computer vision has been doing at some time. They started with this very simple autoencoders, and then there's like all kinds of things such as self-supervision so coming. And I think this could potentially also improve our latent spaces, or maybe optimal transport the constraints.
Yeah. And the other question, about gene programs, I think it is really nice that this is one way of getting interpretability, right? But it seems like you always have this kind of balance of, then you're adding in a whole lot of prior knowledge.
Absolutely.
In order to
get prior knowledge that is also maybe sometimes on the shaky side. Yeah.
Exactly. So it seems like there is this balance, right? So either you want to do everything in some unsupervised way and learn these gene programs. So how do you see this of like making this maybe a bit more?
So this is something that I originally was completely worried, and I actually didn't even want to go initially in that direction. But how I got convinced, and how we convinced ourselves that this actually is doing a good job, is that because now we have a fully data-driven thing with a lot of samples, we can actually add additional constraints. We have a sort of a soft thresholding. We only use this mask in a soft way, and we actually, with an L1 constraint on other components to each of those dimensions, allow it to extend it. And in fact, we could show if we remove an important component to, for example, understand B-cell variability, it would actually need necessarily in this L1 constraint add these additional things. So in my opinion, whatever we use as ontologies of gene groups at the moment, it's manually driven. And as you say, it's going to be too restricted. But if you consistently now add with this more flexible way additional genes in this representation, we can actually update them. And the second aspect that I really like is once we do this reference mapping, we allow with surgery to look for additional components. We can also enrich our dictionary. So I think one of my early hopes when we built these atlases was we kind of need to be able to do better on ontologies, right? We should be able to group things. In a sense, like grouping some genes is the first basis of some type of more complex regulatory motive. And I think in this way, because it's sort of flexible, but it still has the names, I think it's something that sort of maybe hits the sweet spot. You can still sort of tell what this is. It's not just like upregulation of gene X, but it's hopefully flexible enough. And we saw that it doesn't, at least for the integration task, it did not lose expressive.
Mm-hmm. But in some sense, so then you're using the same ones for any cell type. You just have like a collection of programs and you just add them all in, no matter which cell type you're analyzing.
Yes, of course, but of course only some of them are actually then used. So you see like it's not like two dimensions, right? So let's use this one program, and this one is always then off, except for the interferon, and then this is turned up. So it's I guess like in FACS 2D scatter, you can then sort of look where some of them are on, and you can also score this with base factors and so on. Yes, but of course they're initially all shared, and it's got to be this sort of dealing with this.
Yeah. I think you're bringing up something, right, with the invariances. Here you're actually encoding them yourself, whereas, in ML, there should be a way of figuring out something that is a bit more more principled, right? Like convolutions work for basically any of the vision.
Yeah, what's the more principled thing in our case? Yeah,
exactly.
I would be interested in that. I think, as soon as we go to spatial data, it's going to get easier again, because then there should be, it's going to be a graph because it's not going to be on a grid, but, this thing should be then invariant. So that's, there's some old papers, I don't know if you saw this, like network smoothing ideas and so on, where people use PPI and then try to make things sort of invariant under these. It's a bit of a
Great. So thank you very much, Fabian. Really great talk.
Thanks for the nice
So we'll
discussion.
now move over to the second talk. So it's really my pleasure to introduce Samantha Morris. Sam is an associate professor of genetics and developmental biology at Washington University in St. Louis. She's also an Allen Distinguished Investigator and the New York Stem Cell Foundation Robertson Investigator. Her research focuses on dissecting and manipulating gene regulatory networks that define cell identity and applying this knowledge to engineer clinically relevant cell types. So Sam, I'm really excited to hear your perspectives on gene regulation. And again, for everyone who is listening, please type your questions during the talk and we'll have some time for Q&A after Sam's talk. So Sam, all yours.
Okay, thanks very much. Can you see my screen? Okay.
You can. Yep.
Okay, fantastic. Thank you. Yes, so by training, I'm a developmental and stem cell biologist, and the obsession of the lab is how we morph one cell identity into another cell identity. And it's really now coming down to the fundamentals of how cell identity is defined. And this brings us to gene regulatory networks, which I'm going to jump straight into today. So these are the fundamental or master regulators of cell identity. So this is a great place to start as reprogrammers. And we've developed a platform called Cell Oracle, which we use to dissect cell identity via network inference and in silico perturbation. So just to run you through this platform, we use single-cell RNA-seq and single-cell attack-seq to infer gene regulatory networks for each cluster that you would identify in your single-cell RNA-seq dataset. So then you can ask how GRNs change during reprogramming or differentiation or development, and really then ask how transcription factors regulate cell identity. And as reprogrammers, we're very fond of just throwing transcription factors into cells to shift cell identity. So it's also a really fun system to test. So in the first step of Cell Oracle pipeline, we build what we refer to as a base gene regulatory network. So this represents all the biologically feasible connections within the network for a particular species. So we're just talking about prior knowledge, and this is really where it comes into play. So we can do this using a few methods. The main method we use is to take single cell attack-seq data. So you can use data from existing atlases to do this. So we use the mouse single cell attack-seq atlas from Kusanovic. And we use this to identify accessible promoter and enhancer DNA sequences. And we scan these regulatory elements for transcription factor binding motifs. And this creates a list of all potential or biologically feasible connections between a transcription factor and its target gene, so for every cell type within a single species. And then this results in the base gene regulatory network. So in the next step, we need to identify which of these potential connections are actually active for each cell type within a data set. So this is where the single cell RNA-seq data set comes into play. So we use this to build a regularized machine learning model that predicts transcription factor target gene relationships. And from the model fitting results, it presents a certainty of connection as a distribution. And then we use this to remove weak or inactive connections from the base GRN, which leaves you with this cell type or state-specific GRN configuration at the end of your experiment. So step 3, we're often asked how Cell Oracle differs to some of the single cell RNA-seq based GRN inference methods. So the difference here is that we can use this ML model that predicts transcription factor target gene relationships to simulate the effects of transcription factor perturbation. So either knockout or overexpression. Today I'll be focusing almost exclusively on knockout simulations. So how we do this is we essentially say, okay, within a GRN, let's focus on transcription factor X. Let's set the expression of that transcription factor to 0, and then predict the effect on the target genes. So how will the expression of the target genes change? We then propagate this initial perturbation effect through a couple of rounds to simulate the indirect global effects on the transcriptome of this initial transcription factor perturbation. We don't go much beyond this because it could create a lot of noise potentially. But the result of this is a simulated expression matrix that represents the future gene expression values following the specific transcription factor perturbation. And we can project this directionality of future cell identity transitions onto whatever embedding of your choice. And in this case, we're predicting a lineage switch. So at this point, I really want to point out that we don't aim to predict the exact changes in gene expression that arise from a transcription factor perturbation. Rather, we are predicting the differences in gene expression. And this is what allows us to really try to predict these shifts in cell identity. And to walk you through this a little more carefully, I'll show you some specific validations that we've performed and also some novel biology that we found with this approach. So the first validation, I'm picking up this data set from 2015 here on myeloid progenitor differentiation. So this is a fantastic single cell atlas, very continuous trajectories in this data set of cells differentiating from progenitor states to granulocytes, monocytes, and also megakaryocytes and erythroid cells here. So just projecting the wild type cell density here. Also in this paper, the authors included single cell data on CBP alpha knockout cells and found that CBP alpha knockout blocks granulocyte and monocyte specification at these initial progenitor stages. So this is a really excellent ground truth data set to start to understand how cell oracle performs. So how we run through these simulations. So it took us quite some time to realize that we needed to take into consideration the differentiation of our system. So this natural flow of changes in cell identity through the system. So we create a gradient of pseudotime, which represents differentiation or reprogramming or development, whatever is most applicable for your system. We then take the cell oracle CBP alpha knockout simulation, so the Vex field produced from this, and we calculate the inner product between the pseudotime gradient and the knockout simulation. And this produces a cell density prediction. Well, not quite, we're going to get to the cell density prediction in the CBP alpha knockout sample. So just to walk you through these inner product scores here. So the areas in red are the positive inner product scores, and this is where we're predicting differentiation will accelerate because the perturbation works with differentiation. Now, the areas in blue are where we're predicting that differentiation will decelerate. So we blocked, it will revert to an earlier state, or it will slow down because the perturbation effect is working against the natural momentum of differentiation. So we would predict that cells would accumulate in this area. So just to make this a little bit more accessible and easy to interpret, that we use a random walk simulation to simulate the density, so the changes in cell density following CBP alpha knockout. And from these changes, we see that we're predicting a high area of cell density in these progenitor stages and also later in erythroid differentiation. And looking at the ground truth here, we see this accumulation of cells in these progenitor stages and also down the erythroid lineage as well, as expected. So now moving on to a second knockout here, so the CBP knockout phenotype. So our simulated density, we're predicting that we'll have this accumulation of cells in these progenitor states and in the monocyte lineage following knockout simulation. And again, the ground truth agrees with this. So accumulation of cells in this area and loss of differentiated cells down the granulocyte lineage here, which is exactly what we have predicted in this case. So moving on to systematic transcription knockout simulation. So it's great to have this ground truth knockout data to directly compare to. We wanted to perform knockout simulations for all transcription factors within this data set that had an inferred connection to at least one other gene. So this was around about 90 transcription factors in total, and we're focusing on the granulocyte monocyte lineage, and we're also focusing on the megakaryocyte and erythroid lineage here, so kind of splitting up these two lineages. So how do we start to, first of all, perform these systematic perturbations, but then in an unbiased manner, start ranking the transcription factors by their predicted impact, the predicted impact of their loss on each lineage. So to do this, we take that negative perturbation score. So there's a really important point here, that the negative perturbation score represents that when a transcription factor is disrupted or knocked out, that cell differentiation will be blocked. And so we take the sum of the negative perturbation score and we transform this. And then this allows us to rank the transcription factors according to the predicted severity of the phenotype. And we've done this in the ME lineage and also the GM lineage. And just to point out one famous transcription factor here, GASA1, so it receives a high score in this megakaryocyte erythroid lineage. It's a well-known factor in erythrocyte differentiation. And overall, when we take these predictions, 85% of them are supported by published phenotypes. So this is why it's so great to work with hematopoiesis as a system here, because so much is known about it. So now moving into how we can find new biology with this approach. So I introduced the lab as being quite focused on reprogramming cell identity, but this is a project that's taken me back to my roots in development, and a new area for me, which is zebrafish development. So this has been quite exciting. So we took a really lovely atlas of zebrafish embryogenesis that was published by Jeff Farrell in 2018, and we performed knockout simulations for all transcription factors that had inferred connections to at least one other gene, and we did this across the entirety of zebrafish development, or certainly these earlier stages. So this, we boiled it down to 232 active TFs that we performed knockout simulations for, and this has allowed us to prioritize factors, discover new phenotypes and new regulators of axial mesoderm development. So initially, we wanted to take a look at, all of development here, but we ended up focusing in on the axial mesoderm here, just to break down the complexity. Also, the axial mesoderm is a really important patterning structure in early zebrafish development. It splits or it differentiates into notochord and precaudal plate. So quite a well-studied lineage bifurcation here. We first calculate our pseudotime gradient, so this is something we can perhaps discuss afterwards, but it really helps to have strong differentiation gradients in these systems to be able to interpret the phenotypes that result from perturbation. I'm not going to dive too much into the network analysis here. I'm just showing an example here of ranking transcription factors by degree centrality. So this is a measure of how many genes a transcription factor is connected to within a cluster's gene regulatory network. At the top of this list, we find NOTO. So this is a famous regulator of notochord differentiation. And we also find all the usual suspects in notochord differentiation and axial mesoderm formation in general. So this was nice to see. So then, considering NOTO, we just wanted to check everything was working as we expected in the system, that everything made sense. So we ran the NOTO knockout simulation and the cell density prediction. And as expected, we see loss of notochord differentiation in this instance. And we see that differentiation down the precaudal plate lineage is potentially accelerated here. So you're having an accumulation of cells in the precaudal plate cluster. So going back to the 90s here, so this is analysis of the floating head mutant. So this is a NOTO loss of function mutant. And we see here a really nice notochord structure in the middle of the embryo flanked by somites. In the knockout embryos, so NOTO knockout, we see loss of notochord, and really this structure is replaced by somites. So the long-standing conclusion from these experiments was that there's a transdifferentiation between notochord and somites, which is true to an extent, but we were curious about this accumulation of cells in the precaudal plate that hadn't been published before. So we basically recapitulated the Farrell single cell atlas. We produced this high resolution axial mesoderm differentiation atlas. And then we project our floating head mutant cell density onto this atlas, and we can see accumulation of cells in the precaudal plate lineage here. We see loss of cells in the differentiated notochord as well, as expected. And we find, in agreement with our predictions, that we get the significant accumulation of cells in the precaudal plate. And this is really not something that's been reported before because the phenotype isn't obvious. There's a slight phenotype there, but it's not necessarily something you would be able to quantify precisely without single cell technologies. So this was a satisfying result. So next, moving on to finding novel regulators of axial mesoderm development. So this was a challenge. So again, we focused on these negative perturbation scores. So here, focusing on the noto knockout, so we get these strong negative inner product scores at the beginning of notochord differentiation. So again, we take the sum of these negative inner product scores and we transform it. And this produces a prioritized candidate notochord regulator list. And the transcription factors highlighted in red and starred here are the usual suspects, validated regulators of axial mesoderm and notochord differentiation in zebrafish and in other species. And then the factors in gray are transcription factors that haven't previously been reported to play a role in notochord differentiation. So we set out to validate some of these candidate regulators in collaboration with the Solnöcker-Kretzel lab. And Leela Solnöcker-Kretzel performed a lot of these mutant screens in the 90s. So she thought this was an interesting challenge to find something that she states was so exhaustively studied for many years. But some of these factors that were appearing in this list, some of the novel factors were interesting, but we went through another round of selection with these factors. So if I just highlight ZIC2A here, it's expressed within the axial mesoderm, but it's also more broadly expressed within the zebrafish. So we decided not to pursue that as a candidate because it would be difficult to rule out if effects on other tissues were not then having an impact on axial mesoderm development. Likewise, there's other factors in this list that have very few connections within the GRN. And so we focused on factors that had many connections in the inferred GRN. And one of our main factors here is LHX1A. We also tested IRX3A and CBOX. And all of these factors produced phenotypes that were in line with the cell oracle predictions. But I'm just going to focus on one of our favorite mutants here, LHX1A. So prediction of LHX1A perturbation, we predicted to lose precaudal plate differentiation and also to lose notochord differentiation. The cells would accumulate in these early axial mesoderm stages. So we found this predicted phenotype quite interesting. So again, we used a CRISPR-Cas9 based approach to disrupt LHX1A, so to produce these crispant mutant fish. And we find that, so this is data aggregated from 5 independent biological replicates. So we find a significant loss of cells in the precaudal plate. As predicted, and also a significant accumulation of cells in the early notochord as predicted. Again, these fish don't have an obvious phenotype, so this is 10 hours post-fertilization. So this mutant phenotype would never be picked up just based on morphology alone. The fish die 5 days later, but we don't think this is necessarily connected to these early changes in differentiation. Obviously, we've profiled these crispants, these mutants, so we can take a deeper dive into the single cell data. And one of the most interesting findings here is that in our crispants, we have this significant enrichment of anti-dorsalizing morphogenetic protein expression. And it's known that ADMP antagonism promotes notochord formation. So here's a potential mechanism for how loss of LHX1A is resulting in loss of notochord and precaudal plate. Also, to actually show some of these embryos here, so noggin is a marker of the notochord. We see completely disrupted, so consistently disrupted noggin expression in these crispin embryos as well, just for some orthogonal validation. And then finally, how do we apply this approach to less amenable systems, systems that we don't have easy access to, such as human development? Vittoria Bocci approached us earlier this year and said, we have this human fetal striatum single cell atlas, and we'd like to apply Cell Oracle to this. So we have some candidates for medium spiny neuron maturation in the human fetal striatum. So we were able to support some of these hypotheses through simulations, so specifically for a knockout and overexpression of ZNF467 in this case. So just to give you an example of how we can use this approach to generate hypotheses in less accessible systems too. So in summary, I've presented Cell Oracle. So we can use Cell Oracle to simulate cell identity changes in response to transcription factor perturbations in silico. This is based on a new model for network inference. I've presented validations in hematopoiesis and zebrafish development, and this has allowed us to identify new regulators of cell identity. And then finally, I haven't had a chance to talk about this today, but we are finally coming back around to applying Cell Oracle to identify new transcription factors to take us closer to these ideal reprogrammed states that we're trying to capture in vitro. So developing these precision reprogramming strategies. And with that, I'd like to acknowledge the lab. They've been incredibly just persistent over the past 2 years. Also our funding bodies. If you're interested in Cell Oracle and our other tools, then please check out our GitHub page and also the lab website. And I'd be happy to take any questions.
Thank you very much, Sam, for a really wonderful talk. Let me start with some questions. So one of them is about how does Cell Oracle actually predict the effect, or can it predict the effect of other genes beyond transcription factors? So it seems like you have added edges between transcription factors and genes, but how do you take into account other kinds of connections? And for example, could it also predict something about receptors?
Yes, so that would be the next challenge. So sorry, I am trying to. Okay, let's see if I can get my webcam on.
Your webcam is working.
So okay, excellent, fantastic. Yeah, so I think that's the next challenge. So obviously, I make that comment as reprogrammers, we're obsessed with transcription factors as our transcription factor centric view, and so that's useful to us. It's a question I get a lot. What about signaling pathways? And that's definitely the next step in this. So how do we think about the rest of the biology of the cell to make some of these predictions? So it's a work in progress.
But even at the cell, even at the level that you're looking at now with transcription factors, to identify really their effect, how do you take into account? Don't you need to take into account in the network other types of edges other than just between transcription factors and genes?
Oh, yes, absolutely. So that's potentially where we could start with that as well. Yeah, so it's not, so let me clarify here. So we're not just looking at transcription factors, but certainly for the simulations, that's where we're restricted to transcription factors currently. Yeah.
And here there's another question more specific about the method. So how does your method handle conflicts when multiple transcription factors from the same family target very similar motifs?
Yeah, this is a great question. It's something we've looked at specifically as well. So obviously in the base GRN, this is where we account for all potential connections, target genes targeted by a specific transcription factor. It's when we bring in the single cell RNA-seq data. So that's what allows us to distinguish the active connections, and that's what allows us to distinguish which transcription factor is active within a specific context. And this resolves a lot of the conflicts. Now, just to be fully transparent, we haven't searched this exhaustively, but we haven't yet found an example where we can't resolve that conflict. But it's a great question.
Cool.
And can I ask something briefly?
Oh, yes, please go ahead, Fabian.
Sorry, I thought like to for discussion, because I really like this idea of sort of having a simulation forward and then being able to tell what goes. And of course, for this, I understand the transcription factors. Do you think you have a measure of in which situation it works better and which ones not? So, a measure of uncertainty or of noise. So in which case, you might need to extend the model, maybe add something post-transcription and some more complex relations, sort of going in the direction that Caro has, sort of understanding where you have issues there.
This is a great question. So this is what motivated us to perform the systematic simulations. But then we don't have the system, the accompanying systematic ground truth data. So we've tried to use perturb-seq for this, and it hasn't worked great. And this is why we focus more on kind of the classic transcription factor knockout data sets. They're much cleaner, they're much easier to work with. But ideally, say for our 90 transcription factor knockouts in hematopoiesis, we would have the ground truth data sets for that. That will be essential for understanding where cell oracle works great, and then where cell oracle doesn't work so great, and where it needs additional tweaks. In addition, if a transcription factor doesn't have a binding motif, so friend of Gatter, FOG, doesn't actually directly bind to DNA, but it's a transcription factor, in that case, we can't run any simulations. So cell oracle is blind to those factors.
I feel you did a fantastic and great effort to just set up all of these examples. And I think just by itself, this, to sort of to spur on more in the methods community could be a great valuable resource for these people. And maybe also spur on the better development of perturb-seq and crop-seq assays. I got that.
Thanks.
Maybe also in the same vein, also now you're getting back to reprogramming, where of course you need usually multiple transcription factors. So how are you here you always have scores for perturbations on single transcription factors. So how would you go about actually predicting the effect of combinations?
That is an excellent question, because it's something that I didn't highlight in my talk about the way cell oracle is built. So this is a linear model. So I think multiple factors will be tricky. So for single factors, this is working great at the moment, but it would need further development for multiple factors. It doesn't mean that people in the lab aren't trying that and getting interesting results, but Kenji Kamamoto, the postdoc who's the brains behind this, he will get very annoyed with people when they try to do that. So they design all sorts of really interesting epistasis experiments with this, and it's really not what we've intended it for. So in terms of reprogramming, as an excellent example, because we work best within a mapped reprogramming trajectory. So from cell state A to cell state B, and then testing the effect of adding new factors to an existing cocktail. So we don't seek to predict completely new transcription factor cocktails. There's other methods to do that. Likewise, if you're reprogramming and you end up creating a completely new cell state, unless that cell state has been profiled, then cell oracle is essentially blind to it. So we can't simulate into that new identity. So reprogramming is a tricky example. And this is why we've moved back towards development, because it's a much more canalized system. So you can really perturb a lot of different signaling pathways, genes in that context. And you get redistribution of cell types, but you don't necessarily see any new identities emerging. So again, we think it's a much better place to apply a tool like cell oracle.
Great. Okay, thank you very much for a really exciting talk.
Thank you.
So we'll now move to our third talk. It's my pleasure to introduce Jason Buenrostro. Jason is an assistant professor at Harvard University and co-director of the Gene Regulation Observatory at the Broad Institute, where I have the great pleasure to interact with him regularly. Jason's lab develops new technologies that employ approaches across molecular biology, microscopy, bioinformatics in very creative ways. For example, by developing joint single-cell attack and RNA-seq technologies for analyzing how chromatin structure relates to gene regulation, and this in health and disease. So I think we'll hear today a lot about single-cell epigenomics. I'm really excited for your talk, Jason.
All right, thanks, Caroline. I'm going to assume that people can see my slides, but if not, please shout out.
We can.
All right, great. So I'm excited to talk to you today about some of the work we've been doing. As Caroline already stole my thunder and told you all about the things we're doing, I'll try to give you some perspective into some of those areas and where I see things moving towards in the future. So as many of us in gene regulation have thought about for a very long time, as we know, our genomes here, a human genome, as an example, is super long, right? 3 billion base pairs long, and it's hierarchically folded and compacted within the cell's nucleus. And as we think about that structure within the cell, most of the genome exists in this nice, supposedly regularly spaced heterochromatic array, and a smaller subset of the genome exists in kind of this beads on a string-like structure. And as we think about the function of the genome, as we think about the function of different bases in the genome, we usually think about histone modifications that might specify different types of regulatory elements. And there's a long list of these, all of them playing important functions into the role of gene regulation. But over the years, we've leveraged one simple fact about regulatory elements within our genomes, which is that they're canonically nucleosome-free, devoid of nucleosomes, right? And therefore hotspots for transcription factor binding and activity and effects on gene expression. So as the theme of today, we're not just interested in the structure of the human genome, we're really interested in thinking about how cells encode these structures and then decode the information within these structures to then give rise to the expression of different genes and therefore the fates of different cell lineages. So we, of course, try to paint these regulatory activities onto different differentiation systems. So to do that, maybe an assay you're all familiar with, we've been hard at work at developing one particular assay called ATAC-seq. So back in the day when I was a graduate student, we developed this method which simply starts with unfixed native chromatin. You wash in TN5 transposase, which binds open chromatin and insert sequencing adapters. You remove protein from the sample, and you sequence those TN5 insertions on a next generation sequencer. And over the years, we've had success with this approach, profiling rare and rare cell types and states, and now, of course, being used in single cell analysis. So in the original paper we showed, we can move open chromatin maps from 50 million cells there on the top to 50,000 cells here in the middle, and to individual single cells in a paper published in Nature, shown here at the very bottom. And in this process of developing ATAC-seq and then, of course, single cell variants of ATAC-seq, one of the principal things we've learned, or at least I have learned in this process, is that new technology development can be a real engine for biological discovery. And in particular in the context of understanding the human genome and its inner workings and its regulation, continues even today to power understanding of the epigenome and its influence on cells. So with that as a motivating factor, we've been hard at work developing tools to better measure chromatin states within single cells. And over the years, we've developed, of course, droplet-based approaches. Some of these are commercialized now, as depicted here on the left. And most recently, we've been developing methods to do ATAC-seq and RNA-seq in the same single cell, as represented in this paper we published in 2020. So we'll be talking a lot more about that in a moment, but I'll just keep on plowing through to give you all the cool content. But before we dive into all of that, I thought it'd be nice to take a step back and ask, how do we use these technologies, these epigenomic technologies, and what have we and others learned in the field? You've already heard some primers on this from today's talks, but I'll spell them out for you. So perhaps not new to this audience, what might we do single-cell genomics in general? Well, the de novo deconvolution, it allows de novo deconvolution of complex cell populations. We can cluster, we can determine trajectories, we can do a lot to identify cell state with single-cell genomics. Of course, the catalog of expressed genes, as Fabian was nicely motivating, give us really nice insights into the function within that cell altogether. So by looking at our favorite receptor or our favorite transcription factor, we start to annotate these cells with putative functions. So then why then single cell epigenomics? Well, I'll tell you two stories, but I'll spell them out here. We think, and we know, I should say, from prior work, that epigenomics provide insight into cellular potential and memory, early markers of cell type and cell state transitions. Of course, as Samantha really nicely showed, paired analysis with RNA empowers mechanistic models of gene regulation across heterogeneous cell types. And what we probably won't be covering today is an atlas of cell types and regulatory elements would yield much needed insights into the function of non-coding human genetic variation. So just kind of, we work on all three of these facets, but we're going to focus on the first, which is on cell potential. And to start this small story, we'll start first by recognizing Sai, who's a wonderful postdoc in my lab, likely starting his own, well, not likely, will be starting his own lab in the fall. And I'll tell you about some of his work in developing share-seq. Okay, so what is share-seq? Well, it's a method we developed to profile chromatin accessibility and gene expression within the same single cell. Really briefly, it's all plate-based, using split pool indexing of barcodes. And one really nice thing, the fact that we can add additional rounds of barcoding and create barcode libraries on cells indefinitely means that the cost of doing single cell sequencing for us is extremely low. Less than a cent per cell, we estimated, though it can be a little hard to estimate. And also, these allow for extremely large studies of differentiation development. We can profile hundreds of thousands of cells without any much concern about cost, aside from, of course, the sequencing reagents. And in demonstrating this technology, we applied this to a diversity of tissues from the mouse particularly, but one we decided to focus on was on the mouse skin. I won't go into all the wonderful biology in the skin because I'm using it more as a demonstration of the kinds of things you could learn with these sorts of technologies. But of course, I'll show the requisite UMAP, which we're all excited to see. In this analysis, of course, every dot is a single cell. And here on the left-hand side of this plot, shown here, we're showing the clustering of cellular diversity by attack-seq data. Here in the middle and right, we're showing clustering of cellular diversity by the RNA-seq data. The colors between these two plots are matched. And what you can see pretty clearly is we identified 22 distinct cell types. And as you start to look at these maps and try to find specific examples, you'll notice that the colors on the left and the colors on the right are equally separated in these two modalities, suggesting that cell type clusters largely agree between chromatin accessibility and gene expression, which was not a given when we first started this work and something we're excited to do in the really early days of single cell attack-seq. But something that's really motivated the development of these tools altogether, at least in my own hands, in my own work, we're really interested to ask the question, are there exceptions to this rule, cases where you can define a cell type by attack-seq that may or may not be present in RNA or vice versa? So maybe quickly, anecdotally, I'll note to you that there's really clear places where you can find this to be true within the data. So as one example, we know that cell cycle associated gene expression signatures are quite problematic or annoying, if you want to use that word, in gene expression data sometimes, where they can start to mask cellular diversity. And here's just one example of that being true, where looking within our data, we find a cluster that's a proliferative cluster shown here in this dark blue dots, nicely clustered by the RNA. But when we look at the attack signal for that same cluster, we actually find that we can resolve this into two individual cell types as immature and mature cells within the attack. So I hope that motivates this overall question of really getting after this fact of whether or not chromatin and gene expression changes are synchronous during cell differentiation, and may that be the reporting differences in cell state over time. So to do that, it was quite a challenging task, analytical task, but Sai was up for the challenge. And the first thing we thought to do was we have to understand how regulatory elements are connecting to their target genes. Without that connection, it's really hard to make any meaningful conclusions just by staring at UMAPs. So to start in this project, we developed a new approach for mapping distal peaks to genes. And the way we do this is actually quite straightforward. Here, just a summary of this analysis. But what we do is we take the accessibility of any specific peak and correlate it to nearby genes. And cases where that peak and that gene are highly correlated, we can draw a loop, as denoted here in blue, depicting its p-value of that statistical association. Early on, we actually found that loops highly clustered within the genome. And for those of you who know about gene regulation and think about it every day like I do, you'll know about super enhancers. And we actually found that these sorts of analysis really nicely uncovered important super enhancers critical in differentiation of these cells. But it wasn't just that. There's a lot of other really convenient things we started finding when we started doing these peak to gene correlations and associating distal peaks to genes. So first, I'll tell you a quick aside about some really wonderful work that's been ongoing and more has been done since, trying to use functional perturbations to actually map out the effects of non-coding regulatory elements. And in these studies, though, you can argue about power and you can argue about many other things. In these studies, what's being found is that enhancers predominantly target one gene and change expression levels at about 20%. It's also found that functional enhancers are an average of 24.1 kilobases from their target gene, so quite proximal. And this is key and kind of surprising to me, but I think post hoc one can rationalize this in all sorts of ways. But of the candidate enhancers ataxic peaks quote that they tested, only about 11% of those were actually functional. So what that means is that about 11% of distal attack peaks are actually affecting gene expression in some quantifiable way. And as we did our peak gene associations, as depicted a moment ago, what we actually found was really similar numbers. So enhancers predominantly target one gene. Enhancers, as we define them through these methods, are about 24.4 kilobases from their target gene, and about 13% of distal attack peaks associate with gene expression. And of course, to really kind of hammer this down and to validate that this was working well, we found that these distal peak gene interactions that we're computing were actually significantly enriched for orthogonal validation data, in this case, 27 subtle high chip loops. Okay, so now with this approach in hand, we can move it one step forward, and we can just ask the simple question of how many distal peaks correlate to each gene. So I'm going to walk you through this really briefly. So at the very top of this plot, you'll find the gene Notch1. Notch1 has about 46 distinct ataxic peaks that appear to regulate the expression of Notch1. And as you go down that list, you start to find all the who's who of the regulatory factors, right? Including key transcription factors like LEF1, a central node of Wnt signaling, and others as you can start to see. These were oddly reminiscent of super enhancers. I'll say that one more time, but they weren't exactly super enhancers. We found many exceptions to that rule. So we decided to call them something new to not confuse these two disparate definitions. We call them domains of regulatory chromatin, or DORCs for short. In the skin, there's about 857 of them. So using these domains of chromatin, we can start that we're all associated to a specific expression of a gene. We can start to do some really simple stuff like ask if they're highly cell type specific. So to demonstrate that, on each row is a different cell type we identified within our skin data set, and the column is a different DORC, chromatin domain. And we're just showing here, hopefully clearly to you in blue, that a lot of these DORCs are extremely cell type specific, only activating in a single cell type or in a single developmental lineage. So now that we had a definition of DORCs, a definition of chromatin that we're excited to use, that we thought was recapitulating the known biology well, we wanted to look in development and differentiation and just ask the question, what are the dynamics of these gene activation? So to do that, we actually luckily had a really nice differentiation trajectory within the skin, leading to 3 different distinct lineages, as depicted here, starting for original progenitor. And if you just simply plot the chromatin stability for the gene Wnt3, of course, a key gene in differentiation, you'll see a certain pattern where red means on and blue means off. If you plot the gene expression for Wnt3, you'll see a restricted pattern here in the far right. And interestingly enough, as you start to just look at a lot of these case examples, and in this case, you can just subtract these 2 plots here in the middle and left, you can actually find that the chromatin accessibility seems to be opening before the expression of the gene, far before the expression of that gene. And we hypothesized that this might be associated with the lineage priming in this particular point, right before the cells undergo terminal differentiation. So to kind of spell this out a little bit more clearly and continue to investigate this process, instead of looking at plots, we decided to formally order the cells in pseudo-time, as we all have done before. And with those analysis, what I can show you here now is the exact stepwise events that lead to the expression of the gene Wnt3. So as you might expect, here at the very bottom, we can now order cells in pseudo-time and see that the last thing to happen is the mature RNA expression as denoted by the number of counts in the exon. Right above that, you can see the intronic counts of that RNA turning on, which is a good measure, of course, nascent transcription. And even before that, you see the activation of the promoter showing up here as in pseudotime turning on just before the nascent transcription. But as we peel back away from this core regulatory part of the gene, the promoter, and the downstream expression, we start to see that individual enhancers or distal regulatory elements, if you want to be more vague, start to turn on far, far before the expression of the gene. And you can, of course, order that now into enhancers that seem to turn on quite early and enhancers that turn on quite late. And if you find this sort of visual a bit unsatisfying, I'll give you one more. So here we can actually do this as a movie. I had to twist Sai's arm, but I was successful to actually make this into a movie. But here we're going to see the cell shown as a kind of a pseudo cell, I should say, shown as this blue circle, and you're going to see it move and roll down differentiation to different lineages. I'm going to play that one time, and you'll see the chromatin opening up and the downstream expression coming on just a bit later. So what we found is that, in general, enhancers turn on before promoters, and promoters turn on before gene expression, giving us a general model for gene activation development. We wanted to go one step further than just describing the activity of events that might happen in sulfate decisions. So to move that one step further, we wondered if we can use this information as a way to predict lineage relationships, right? So as a simple question, if you have a stem cell shown here in green, can you predict whether it might prefer to go to, quote, lineage 1 in teal or lineage 2 here in yellow? And we reasoned that these dorks or these enhancers can be very useful to predict sulfates. We're inspired by work for demonstrating RNA velocity, and Fabian mentioned this a bit as well as the long-standing work in epigenomics annotating K4 monomethylation, in particular associated with gene priming, kind of a known fact, a stage of gene activation. So to do that, I'll spare you all the technical details, but I'm happy to get into them. In short, what we simply did, I'll do a terrible job explaining it too. We correlated each cell in chromatin space and dork space to its most similar cell in RNA space. And based on the correlation, presuming that chromatin always turns on before the expression of the gene, we reason that the arrows that we can connect those two kind of matchings would always point to the direction of differentiation. Doing that, we found that it did. So here's the same data set I mentioned a moment ago, and now we can see this imbued with arrows pointing largely in the direction differentiation is denoted by pseudotime in the coloring here. And importantly, something I don't have as much time to get into, we also found that there was prime progenitors within this system, something that we can validate with prior literature using kind of lineage tracing approaches that have been done carefully over time. So summarizing this part of the talk, chromatin and RNA-defined cell types are generally, though not always, the same. And when there's exceptions, here we're finding that chromatin change can precede the expression of gene expression, right? Precede RNA change. And of course, can be even earlier predictors of sulfate decision, even more than the nascent expression of genes. What we're also kind of getting at with some of these technologies is that, we'll hear more about that in a bit, but integration of ATAC and RNA and doing these at scale can elucidate regulatory mechanisms promoting gene expression. Here, the definition of dorks and distal peaks in the target genes. And what we're excited about doing forward, doing a lot more going forward, though maybe this will kind of be more in the hands of expert computationalists, is to improve the computational tools for lineage and regulatory mechanism prediction, as you've heard already. There's a really interesting intersection, I think, between these two areas. Okay, so moving on to the second part of the talk. We come back to these core essential questions, and now I'll try to convey just a short vignette of unpublished work that I think might speak to this well, which is how can we use the paired ATAC and RNA data to empower mechanistic models of gene regulation across heterogeneous cell types. So quickly motivating this second half of the talk, we of course think about structures within cells a whole lot. We're very obsessed with them as biologists, I would say, kind of subdividing things into finer and finer granularity. And at the broadest scale, right, we can think about chromosomes and chromosome bands and even chromosome subbands, right? And even below that, we can think about topological domains as we think about gene regulation, right? These are these regions of the genome that are interacting and are kind of usually as gene regulation blocks. And even a substructure within that, you could think about distal peaks in our target genes, as we just described a moment ago with that share-seq work. And perhaps even finer than that, is there structure, I guess is the question, but is there functional substructure even within an individual regulatory element? And I think it's fair to say that at this last resolution, things are generally underexplored. We tend to think about a tax-seq peak or a enhancer as a single entity with a singular function, but we very, very rarely try to dissect a single peak into requisite components that might lead to its function. So what we've been wanting to do now with single-cell tax-seq data, and one of the things that we've always wanted to do, but we're always limited in a few different ways, which I think we now have solved, is to try to footprint the bindings of TFs. So at a large scale, regions of the genome can be opened or closed, right? And that's read out by the presence or absence of nucleosomes. But at an even finer scale, about a 10 base per resolution, transcription factors are binding, and here's the crystal structure of that event. And we reasoned, and this has been shown by us and by others, that if a TF is bound, it might actually stop transposition from happening at that very small window, and we can read that out in the tax-seq data. So while we've done this before in the bulk, we wondered, can we resolve DNA footprints now at single-cell resolution using some of these large-scale single-cell genomics tools that we have at our disposal? And this is work led by Simon, postdoc, which you've heard just about, and Jan Hu, who's a wonderful graduate student, probably listening to the talk right now. So I'll really briefly go through this, but we're also obsessed with hematopoiesis because it's a wonderful system, as Sam said, for understanding how transcription factors lead to lineage fate. And in this process, we've decided to generate a large-scale data set covering about 1 million human bone marrow cells, representing about 1 billion reads aligning to the human genome. And if you haven't thought about hematopoiesis as much as Sam or I have, I guess, the first thing you have to know is that the hematopoietic stem cell in green, it gives rise to all the lineages, and you can see that bear out as you start to see these arrows emanating from the central point source in green. So we did share-seq. And then to footprint, we then, any one cell is way too sparse. So what we have to do is aggregate similar cells. So we build the K-nearest network graph, and we start to aggregate quite similar cells in this analysis. So now that we've done that and have our little pseudo clusters or pseudo bulks defined, we can start to build footprinting methods and tools. In brief, I'll show you an example of what one attack-seq peak looks like. So generally, when we do this analysis and what's part of most standard workflows, what we usually do is we smooth the data with the 200 base per kernel. So what you do is you take this 200 base per kernel and you just smooth as a sliding window across the genome, and that represents those peaks that I showed you earlier and ones that you probably have seen represented in most papers. And that can be shown in this as this black line here. But if you just simply take a smaller kernel, note this only works when the data is really, really deep. But if you simply take a smaller kernel and do the exact same operation, what you'll start to find is that there's actually a lot of structure here, where you can see really nice bigger dips and more focal dips occurring within the data. So now the challenge was to take this higher resolution data, do all sorts of bias correction to regress or remove out kind of preferences of the transposes itself to systematically and statistically compute these dips. So using this tool we built, we haven't named it yet because it's unpublished, we're able to actually resolve the positions of transcription factor footprints within here one individual peak shown in red. Now, you shouldn't stop just there. You shouldn't stop with one single peak and one single pseudo bulk or cell. What you should do, and what we did do, is to repeat for all cell neighborhoods in the data. So you can do this for every single unique region of cell state space that you cover within a single cell data set. So now every row is a different pseudo bulk, unique state space, and every column is a different position along that same peak. And each of these rows are colored by the cell type that we identified that as. And what you already see is that there's actually diversity in structures across different cell states where you can actually hopefully make out that there's essential stripes in the middle that denote a few transcription factor binding, but of course these other stripes that tend to occur at the flanks. As we stared at many of these, not just this one, we started to realize that things were also apparently occurring in blocks. Here's one where you see the gain of footprints for this coherent block within monocytes. Here's another one. You see almost extension of that to the far left. Here's another block here in the far right, and here's a fourth block. And if you actually measure the size of these blocks, you can already intuit this. What's the length of a DNA on a nucleosome? Well, it's 147 base pairs plus some, and you can actually make out that these are about exactly the positions of nucleosomes flanking these regulatory elements. So to actually kind of more systematically look at some of these placements of these regulatory factors, we moved our transcription factor footprinting tool from 10 base pairs, as shown here on the left, capturing the bindings of TFs, increasing the window size, to now positioning of nucleosomes by just widening that window. And this is shown in gray, where regions, of course, more gray is the positions of those nucleosomes. Now, what we can do is just simply kind of in this multi-view way, overlap these two signals, the TF footprints and the nucleosome footprints, we can call them, and find that where nucleosomes aren't, TFs are. Perhaps a not surprising kind of finding, but at the same time, you can see it beautifully play out here as this nucleosome, of course, has to get out the way so that transcription factors can actually find their way to these regulatory sequences. And what I haven't had a moment to really kind of instill upon you, we also, of course, have the gene expression data from all of these events. And when we actually find, as denoted here in the top, very top, as we correlate these footprints to gene expression, we actually find that this particular flanking region here on the right is most associated with productive gene expression, suggesting that these center stripe factors are, what should I say, priming or establishing open chromatin, while these rightmost transcription factors actually leading to productive gene expression, separating gene activation into different discrete stages. So in this process, we think we're finding all these really fun dynamics over differentiation development. We're looking a lot more into this data. As I said, this is unpublished. But this goes back to some of the reasons why I got into this whole area to begin with, kind of inspired by what Sam was saying about development. Well, I'm most interested in understanding how regulatory factors work together or sometimes even compete for access for DNA. And the canonical model, as we know, is transcription factors need to compete with nucleosomes for access to regulatory DNA. And we're starting to see that play out really, really beautifully as we start to do these footprinting studies at single cell resolution. So summarizing the last part of my talk, I'll say a few things, which is single cell epigenomics at scale enables transcription factor and nucleosome footprinting. That's something that's really exciting and I think will be emerging in the next few years. Nucleosomes move and slide during development, exposing DNA for TF binding. It'll be interesting to think about the exact timing of those events and who instructs who. Integration with RNA-seq uncovers footprinting events essential for gene expression. It's already been fascinating to uncouple these two events, something that leads to productive opening versus something that leads to productive gene expression. There'll be, of course, factors that are responsible for one or the other. And then ongoing, we think that these transcription factor footprints may reveal a functional basis within regulatory elements, and this is ongoing work, and we're excited to find more opportunities to kind of maybe even collaborate with others in the space. I believe this might be a really great way to annotate the function of non-coding genetic variation, which we're excited to do. So with that, I'll thank Sai and Jan, who really drove this project, and the whole lab depicted here, having fun before pre-pandemic days. Our many collaborators, specifically Yache Su and Aviva Gev, who've been instrumental in developing the original share-seq approach. Our institutions, Harvard, HSCI, Harvard Stem Cell Institute, and the Broad Institute, as well as the GROW, as well as our diverse funding sources that make this work possible. With that, I'll take questions, and I can't see your faces, but hopefully we'll soon.
Thank you very much, Jason, for a wonderful talk. So in terms of questions, so I'd like to first start with, you have very nicely shown how ATAC-seq, the peaks are often ahead of the RNA-seq peaks. So it seems like you may not need RNA-seq at some point. So can you talk a bit about the things that you can, so you said a lot about going in one direction. Can you maybe say a bit about the things that you cannot measure from ATAC-seq directly about RNA-seq?
Yeah, so I guess you're saying a few things. Like, do we need RNA-seq data? Actually, you could do a lot with just the ATAC data. But at the end of the day, I think that gene activity or gene expression can come from, a few different mechanisms. And to really kind of tease that apart and to really, if the goal is to understand the determinants of gene expression, I think you got to measure it, So I think that is the goal. Though being an ATAC-centric person, I think there's been times when I've almost booted gene expression analysis from my own workflows. But I think if the goal is to understand the function of open chromatin, I think having the gene expression output would be really essential. And then I think maybe adding another piece, if the goal is to understand cell state, I think there's lots of cases where the cell might carry with it information of a primed cell state, but the only way to really know if it's been committed into that cell state is to look at the gene expression. And we do see lots of examples of that as well, though it's kind of hard to capture in this particular study in a coherent way.
Cool. And in terms of connecting it back to Fabian Theis' talk on looking at latent space embeddings and multi-domain integration, here you have, of course, nice pairing, right? But if you're looking at the variable, like how much of the variability is captured, how much of the single cell variability is captured with one data modality as compared to the other, do you see big differences?
Yeah, that's a really wonderful question. It's something we really wanted to get at in a quantitative way, but I think as you can appreciate, it's probably very hard to be quantitative. And you can do a few things to try to get at that. One of them is like kind of distill down to regulons and think about kind of where variation might be coming from and ask about membership into those things. And in those efforts, though none of it published, is just kind of our own kind of queries into this. What it seems to be is that anytime you see a chromatin change, you see an RNA change and vice versa. That seems to be almost always true. But there's a lot of warp in that translation, where there might be something that's a lot of RNA change, but with very little chromatic stability change. There's kind of this translation that happens where maybe a subtle change in chromatic stability might lead to a very large change in gene expression, and also the vice versa. So things like lineage priming, lineage potential changes in that might actually not lead to very drastic changes in gene expression, but might be really kind of, I guess if you want to use the word, inflated or perhaps emphasized in the chromatin state. And I think that's the really fun part, is that perhaps just by looking at gene expression, we're kind of a little bit biased towards certain types of programs. One is cell cycle, as an example I used. But I think if you focus on chromatin, you actually see really potent signatures of lineage kind of happening even before the expression of the gene. Though if you stare, you might find a little bit of expression of those genes too.
Cool. And in terms of your dorks, and maybe also thinking about perturbations, so if you do perturbations in those areas, so how robust is it, right? So how many, if you perturb just one element in there, how many different ways does the cell have to rewire around it, or do you actually have to perturb all of them together?
Yeah, so that's a really wonderful question. So I can mostly talk to you about some of the kind of fields work, not just my own, right? Because these are also super enhancers. And there's a few interesting things in discovery of these super enhancers. What was found is that they're also hotspots for genetic variation. And you might wonder, like, how does that make sense, right? These things, there's many elements. You would think it would buffer out pathogenic genetic variation, but it turns out to be the case that it might actually not be, that might not be true, actually. There's more opportunities for there to be a pathogenic variant in these larger domains. So in perturbation studies that people have done of these elements, it looks like there's often hub regulatory elements that are particularly important for kind of licensing the whole domain. So there might be various numbers of regulatory elements. It might be more tuning kind of effects where if you mutate or knock them out, you might get a subtle effect, but then there might be also hub elements where if you knock out that element, the whole thing kind of gets destroyed. And we see that play out in some ways. When you look at these movies, and if you kind of stare at the one I even showed, you'll see like this central peak that shoots up really early at a really fast rate. And then like right behind it, the whole domain gets formed. So one might hypothesize that we don't have data for, that these might be kind of an essential beginning for that whole element to awaken.
Cool. Very, very nice. Thank you, Jason.
Awesome. Thanks.
And with this, let me thank all three speakers for like really, really wonderful talks, providing very different perspectives on, important biological questions around gene regulation, technological developments that allow us to, ask new questions about gene regulation, and also, of course, computational methods that are required in order to analyze all of these amazing data sets that are coming out. So with this, I would like to end this really amazing session. So thank you all for being part of it. I hope this gave you a lot of food for thought and a lot of new problems and a lot of motivation to actually get excited about this field and think about all these questions that are maybe now approachable in this area of, better understanding gene regulation. So with this, happy holidays to everyone, and thank you all for being here.